Hi, I'm Glenn Gaudette. I'm at Boston College, professor and chair in the Department of Engineering, and I wanted to share with you some information on what's commonly referred to as lab-grown meat. So in the next series of slides, I'll kind of give an overview of the field and some specific examples. So here we go. The outline, I'll start off with some definitions I think might be important. Then we'll talk about why do we need lab-grown meat? What's the purpose? After that, we will talk about how to make lab-grown meat, kind of the ingredients, I would say, that go into lab-grown meat. And after that, we will talk about problems limiting commercialization. So how come we can't get this on the shelves today at our supermarket, our food store, or at our favorite restaurant? And then share some other considerations. You'll hear me refer to lab-grown meat or cultured meat or clean meat. These are various names that have been used at different points in the field. And it really refers to meat grown in laboratory-like conditions. Cellular agriculture is kind of the field that is above lab-grown meat, and it deals with growing agricultural products from cells rather than whole animals. So this includes lab-grown meat, but it also includes other things also. And so we can think about, can we, for example, can we grow milk? Do we need a cow for milk, or can we grow it? There are companies that are doing that, but we're not going to talk about that today. That's kind of beyond the scope of this discussion. Media is something my students refer to as Gatorade for cells. It's the solution that keeps the cells alive and keeps them viable and functioning. We can add different things to the media to change the condition of the cells. So, for example, we could induce what we call differentiation in the cells. And so differentiation is essentially the process of changing a cell into a specific type of cell. We could add different solution to the media that will cause the cells to proliferate or double and multiply. Generally, most cells will either proliferate or differentiate. But once a cell has differentiated, it's hard for it to start proliferating again. Not always, but in most of the cells, especially in the cells that we're going to be talking about in the field of lab-grown meat. Scaffolds is the substrate or the material that's used to grow cells on. And a bioreactor is a device that is used to grow cells in. And so we'll talk a little bit more about these here in the next few slides. So why do we do this? Well, we know that the world's population continues to grow. It's expected to grow to about 9.7 billion people by the year 2050. How are we going to feed all these people? That's a big question. And a lot of the people in the world are starting to enjoy meat and want more meat. And so that means we're probably going to have to increase our supply of meat. Well, that means we're going to need more land. We're going to need more water. They're going to need more resources, energy resources, for example. And where are they all going to come from? That's something we've got to think about, especially regarding the sustainability of our earth. Turns out that animal agriculture is responsible for about 14.5% of all man-made greenhouse gas emissions. And that number will vary a little bit depending on exactly how you're defining animal agriculture, but it's a very, very significant percentage. There's a lot of methane that's produced, there's a lot of nitrous oxide that's produced, and it's something that we need to think about. In addition, about 70% of all agricultural land is used for farm animals. So if we're talking about increasing the conventional meat industry to produce more meat, we're talking about more land, we're talking about more water. Where is it all going to come from? So we can, and I believe we must do better collectively together. So how can we do that? Well, that's where I think lab-grown meat comes in. Lab-grown meat, clean meat, is different than plant-based meat. If you've heard of the Impossible Burger or the Beyond Burger, those are plant-based meats. Those are different than what we're talking about here. Cultured meat is actual cells. It's actual muscle tissue. It's made from animal cells. As I mentioned earlier, you may hear cultured meat called clean meat. You may hear it called lab-grown meat or even cellular meat. And so how do we make this? Well, it largely grows out of the field of tissue engineering. And in tissue engineering, we try to recreate organs and tissues for humans who, for various medical reasons, need new tissue. My lab in the past, for example, has worked on trying to regrow and regenerate heart muscle tissue. And so lab-grown meat has really grown out of that tissue engineering labs and industry. So what does it take to produce meat? Well, it takes cells, it takes media, and it takes scaffolds. We put them all together in something called a bioreactor, and out comes edible meat. Of course, it's a little bit harder than that, but that's essentially what it takes. And that's the way I've kind of tried to break up the talk a bit. If you look on the right here, you'll see hopefully some movement. Those are actually cells that are contracting. And so when you look at the kind of movement, those are muscle cells that are actually contracting. And those muscle cells, in this case here, were grown on top of a decellularized spinach leaf. And so that's work that, excuse me, in the past we've done. And so we've taken human heart muscle and grown it in the lab. Can we take cow skeletal muscle or meat, for example, and grow it in the lab? And so we've used similar techniques to do what's shown here on the right as what we're proposing to do and what the industry is currently proposing to do. So when we talk about cells, what type of cells are we talking about? Well, if we think about the type of cells that make up meat, they're mostly muscle cells or myocytes. There are fat cells in there, and there are blood cells. And so the process of essentially the way muscle grows is shown in the top center in these panels 1, 2, 3, 4. We have what are known as satellite cells, which are shown in red over here and here. And when muscle is injured, as schematically shown by this kind of cut down the middle of this muscle cell, for example, signals are given off to the progenitor, to these satellite cells, which then proliferate. So they grow and increase in number. And then those cells migrate or move towards that injury, and they fuse with that existing cell, that existing myocyte. And so that's shown in phase 4 here, slide 4. So that's essentially the way the muscle grows in our body. And so can we do something similar to that in vitro or in the lab, essentially? What we've done, and if you look closely at the bottom, especially at this end here, and I'll move that laser pointer out of the way in a minute, I'll leave it right there. Hopefully you can see that cell twitching or actually contracting. That's a myocyte, again, a myocyte being a muscle cell. And I guess one thing I should point out too is that skeletal muscle cells or myocytes are multinucleated, which means that they have multiple nuclei in a single cell. And that's what's kind of shown schematically here in 1, 2, 3, and 4. The red dots, well, the black dots are the nuclei inside the red cells, and they kind of fuse together with the big cell shown up here in number 4. What you can see down here is a myocyte that we grew in the lab from satellite cells that we isolated from cow muscle. And so you can hopefully see that contraction. You can see the long slenderness of that muscle cell, and it contracts and stimulates, showing that it's actually a functioning cell. So that's a myocyte muscle cell. When we're thinking about cells for lab-grown meat, there's a whole bunch of things we could think about. One of the things we might want to think about is what source, what animal do we want to use? Do we want to use cow? Do we want to use pig, chicken, fish? There's a whole bunch of different programs out there using a whole bunch of different types of cells. Some of the questions you might want to ask are, do we want to genetically modify these or not? So we can take a cell and then genetically modify it so that it will proliferate over and over again. Right? That might make it cheaper, but it's a genetically modified organism, a GMO. And so is that something we want? Sometimes these cells can become non-adherent, dependent. And so that means they don't have to grow on, they don't have to hold on to something in order to grow. That's something that can be genetically modified again. And that has an advantage if you're trying to grow these cells, especially in a bioreactor. You don't need a scaffold. But again, it's a genetically modified cell. Some of the other things we want to think about, especially as we start moving these cells towards the industry and producing them, what are the quality control markers that we have? How do we know that that cell that I show you in the center of the slide here is actually a good cell and is something we would want to eat? How do we know when it's done, essentially? What's the protein content? What's the amino acid content? Those are some other questions that we might want to ask and have some quality control metrics on. So many different questions we can think about. In addition, in just growing the cells or getting the cells, where do we want to isolate them from, right? Where on the animal do we want to take them? How old should the animal be when we take them? How many cells do we want to take? And so, for example, if we have a cow, when we take a small biopsy, small chunk of muscle, and we isolate those satellite cells and we grow them, will they grow faster if we get them from a young cow compared to an old cow? Will they grow faster if we get them from one part of the body or another part of the body? All these are questions that currently exist that we're still thinking of. Well, what about scaffolds? Again, moving on to the next in the list here, we talked about cells. Let's think a little bit about scaffolds here for a minute. Most of the muscle cells are what we call anchorage dependent. So that means they need to grab onto something. They need to hold on to something or else they're going to die. And so that's the scaffold. Again, we could genetically modify them so they don't need to do this. But then it's a GMO food. And they still may need a scaffold even once they're genetically modified, especially when they begin to proliferate. When they begin to differentiate. We can proliferate them generally when they're not attached. But when we start to differentiate them and turn them into the muscle cells that I showed you a couple slides ago, that's when you generally need a scaffold. So some of the work we did some years ago, we took a spinach leaf, like you see here in the slide, and this is a time-lapse video that took place over 5 days. We stripped away the spinach cells, the plant cells, and we're left with this cellulose scaffold. That cellulose scaffold, then we were able to grow muscle cells on. And so if you recall from an earlier slide, I showed you heart muscle cells contracting on a decellularized spinach scaffold. Well, it's a scaffold that was created very similar to the method that I just showed you here. If we're thinking about growing or using these scaffolds for lab-grown meat, we need to be able to mass produce these scaffolds. We need to produce them in a large quantity. The previous slide, we actually stuck a needle into the end of the leaf and used soap essentially to strip away all the plant cells. That's not going to work if we want to grow these scaffolds in a large number, right? We can grow the spinach, but then we can't take the time to put a needle in each spinach leaf. So what my lab's done in the past is essentially taking spinach leaves and soak them in soap, in the soapy water. And after a few days, they become clear and translucent, like you see here, and on the blow up on the right. And if you look closely, hopefully you can see some of the veins still in the leaf. And then we could actually lyophilize them, essentially freeze dry them or store them in water until we're ready to use them. But we don't have to just use spinach. We could also use different types of edible plants. We could use jackfruit, and so we decellularized jackfruit. One of the things that we often think about when we're using growing cells, I mean, is we grow them in a bioreactor. They sometimes need to spin around, and we need to use different carriers for the cells. And so on the right side of this slide, an example of a scaffold is decellularized broccoli. And so in the top video, you can see the broccoli kind of spinning around in that jar, and down on the bottom, you can see the fresh broccoli. And then once it's been decellularized, what it looks like. And so here's a video of essentially what the process looks like. Here's the cellularized, the regular broccoli florets. We can put them in conical tubes, these 50 ml conical tubes, or we can put them in a big beaker like you see here on the left, and then just slowly spin them around in a solution of soap. After a few days, if you look on the right, you can see, for example, these 50 ml conical tubes. And in the one with the green cap, you can see the decellularized broccoli florets. And so that's essentially what they look like. We also need to think about the media. So again, we talked about the cells, talked about the scaffolds, but that Gatorade for the cells, the media, that carries the oxygen, it carries the nutrients that the cells need to stay alive, to stay viable. It also takes away a lot of the metabolic waste. So when we think about the cell media, we can use several things, or we can add several things that cells need to survive. Glucose, for example, amino acids, proteins. We can also think about adding growth factors, and they could be useful for the cells to proliferate. They could be useful for the cells to differentiate and turn into the muscle cells. Quite frequently, we also think about using serum. We don't know exactly what is in the serum, but we know that it helps the cells grow. And so there are some serum-free solutions out there, some of which have been used to grow meat. The ones I've tested don't seem to work as well as the normal serum. The problem is we get that serum generally from animal blood. And so if we're trying to reduce our reliance on animals and the number of animals we need to get meat from, we're not going to be able to get the serum, right? And this media is generally very expensive, and it's one of the things that drives the cost of lab-grown meat up, and significantly up. You know. In the right-hand side here, you can see a 50 ml conical tube filled with some media. In this case here, it's pink, which is just a pH-sensitive dye, and so if the pH changes, it generally turns more to a yellow color, but a good pink media in our lab generally means it's doing pretty good. So one of the ways we can think, and we've thought about recycling or bring down the cost of the media is really through recycling the cell media. And in this case here, what you see on the left is we've incubated media that has what we call spent media. So we grow the animal cells in the media, and then we take the media and we grow algae in there. And the algae will take up the CO2, produce O2, but it'll also take up ammonia. And so on the right-hand side here, we're looking at the levels of ammonia. And so in growth medium, and so the growth media, the solution that we use to grow the cells in, it starts off around 0.7 millimolar per liter. As we grow our cells in there, our animal cells, we see an increase in the ammonia level, which then has a negative effect on the other cells, and it slows down their growth. And so that's why we need to change out that media every couple of days. Well, if we treat it with algae, we get the green line here, the green box, and we see that the ammonia concentration decreases actually below the original level. And so now we can use that to grow cells again. And that's kind of shown in the middle here with two different types of cells, but basically showing that when we take this media that has been conditioned by algae, we could then grow cells in there again. And so, and we can remove the algae through this process on the left here, just a sterile filtration system, basically. You can see the algae on top there once we're done. Um, and so when we take these cells and when we take the media and we take the scaffolds, we put them all together in a bioreactor. And so the bioreactor provides an environment for cell growth. And if you look here at the this slide on the right, this schematic diagram here is a simple bioreactor system. It has a bar down in the middle here, which slowly rotates, and as it rotates, it kind of moves the nutrients around. It takes the ammonia we just talked about, and will move that so that there's not a high concentration of that around the cells, which can affect the cells. In addition, we could essentially pipe oxygen in there. And so that's the bioreactor. One of the problems with bioreactors, though, we need to understand the oxygen distribution inside there. And so colleagues have been working on computational fluid modeling. And so some CFD models to see what is the optimum speed that we should turn that bar at. How much solution should we have in there? What happens when we have more cells or fewer cells in there? And so this here kind of gives you just a general idea of some of the things that are involved in that. And so trying to get a computational model of what's going on inside that bioreactor so we can try to optimize the RPMs and some of the other things that we can control in these bioreactors. So another thing we can think about when we think about bioreactors is a continuous flow system. And so when we talk about the media here, do we want the bioreactor and the media inside there to continuously flow? What happens as we produce meat? Do we want that to continuously come off? Or do we want to close essentially that proliferation reactor, which is shown here? And then once the cells have proliferated and it's full, then we take that and it's done, right? Single use, we call that a single use bioreactor. Or do we have a continuous bioreactor that continues to basically produce meat over and over again, right? Or is it a single use where we've got one tank, it fills up with meat, done. Now we take that tank and we put it to the next, onto the next stage, which is generally the differentiation reactor. So this schematic here talks about Just some of the one example of a potential bioreactor system. We have a proliferation reactor, and so the cells are growing and proliferating, and the number of cells is increasing. They can go through a quality control, and they could then go into another reactor, another bioreactor, in this case here, differentiation bioreactor. So now they're turning into the muscle cells we want. Again, they could go through another quality control checkpoint, and when they're full, they can go off to packaging. If they're not, they can go back into the reactor. Some of the things we've got to think about, we want a minimum user interface, and so we don't want to have too many people there running this system. What kind of sensors are we going to need? We want to minimize the shear. The shear is as the blades are spinning around, it's creating this stress, this shear stress on the cells. How much do we want to proliferate them? How much do we want to differentiate them? How do we handle the fluid? How do we get it to go from the proliferation reactor through the quality control checkpoint into the differentiation reactor? How do we prime this? Is it scalable? Those are just some of the questions that we need to think about as we're trying to think about bioreactors. There's many problems that are limiting the commercialization, though, of cultivated meat or clean meat. First, one of the big problems is it's difficult to do. Right now, it takes a really skilled workforce to do this. So most of the people working on this have at least a bachelor's degree, and a lot of them have PhDs. So it's expensive. All the things we talked about, the media, the cells, the scaffold, those costs add up, and so it costs a lot of money. In addition, labor is expensive. If we're hiring people that need a minimum of college degree, and a lot of them have PhDs, then labor expenses are going to be pretty high, which leads to high cost of lab-grown meat. We don't know how the consumers are going to react to this. And so will they eat this? Will it be of interest to them? Will it taste good? Will it give them the nutrients that they're looking for? And finally, regulatory approval is needed in the US. We have an FDA, USDA partnership. There are a couple companies that have recently within the last, I guess, about a year ago, been approved to sell lab-grown meat, essentially. And when we also think about the entire process, and that's kind of put together here on the right, we think about the cells, and so where are we going to get them in the cow? We think about the bioreactor system here in the middle. Can we reduce the cost of media by different systems? How fast do we spin the blade? We need to think about computational models. What type of scaffolds are we going to need? How are we going to get them into the system? That's a lot of the engineering, but we also want to think about our workforce and how can we work with the farmers today, right? This is their livelihood. And so can we involve them in this process instead of taking, the livelihood away? Can we work together with them? And that's something I think we should think about in the future. When we think about the process, is it really as simple as adding cells and media and scaffolds together in a bioreactor? No, there's a lot more to it, and some of those things are outlined here. We got to think about, again, we talked about the donor. What's the doubling time of the cell? So how long does it take to proliferate? Is it stable? How long does it take to differentiate? Are they anchorage dependent? Do they need to grab onto something? Bunch of other questions we have for cells. In the scaffolds, what do we want to make that scaffold of? What does it look like? So what does that surface topography, the top of the surface, look like? What's the surface area to volume ratio? Is it edible, or do we have to remove it? I showed you one type of scaffold, but there are many, many different types of scaffolds, some of which are not edible. And so, do we want to use a non-edible scaffold and then grow the cells and then take them off of the scaffold? That could be done also. When we think about the media, what's in there? Can we use serum-free media? Do we have media that's designed to proliferate the cells, to differentiate the cells? What's the cost of it? It's very, very expensive. And then we have some things that overlap, right? What's the optimal population when we're talking about media and cells, right? What's the best media to scaffold ratio? We can think about, the media, the cells, the scaffolding all together, right? What's the optimal cell density? What's the growth like? Logarithmic? Is it linear? What's it going to take? Exponential growth? We don't know. We assume it's going to be exponential, but there's a lot of things that are going to affect that. How will the cells attach to the scaffold? A whole bunch of different concerns that still exist. And really, the field is just starting, and a lot of work still needs to be done, but I think we're headed in the right direction. Here are a few different materials for reading if you want. The first one is just some of the work we've done from our lab, talking about spinach. The next one really talks about bringing cultured meat, lab-grown meat to the market. And it looks at it from an economic point of view, a technical point of view, a social point of view, a regulatory point of view. And there's a lot involved there. And then a very recent, about a year ago, article talking about lab-grown meat and the science of turning cells into steaks and chicken nuggets and other things. So thank you for listening. I hope you enjoy this. And I happy to take questions. If you have questions, you can email me. Again, my name is Glenn Gaudette. I'm at Boston College in the Department of Engineering. My email address is gaudettg@bc.edu. Thank you very much.